introduce yourself and tell us a little bit about yourself and your background?
All right. Well, my name is Scott Bowdridge. I am a associate professor of food animal production at West Virginia University. I grew up raising sheep in Southern California in the middle of the desert in between LA and Vegas. So my family raised Rambouillet and Oxford sheep there. I went to college at Chico State in Northern California, taught high school ag for a couple of years down in Central California, then moved out to Maine. And I was the shepherd for a nonprofit rare breeds farm in mid-coast Maine. And then I got interested in parasite resistance and parasite problems in sheep in general. So I worked with some guys that were doing the Katahdin hair sheep project. They were doing some upgrading of Katahdin sheep in Maine. Did my master's at the University of Maine, then went down to Virginia Tech to work with Dave Nodder and did a PhD there before starting here. So my research interest and my focus is on improving parasite resistance in sheep and kind of understanding immunological mechanisms, selection for, so some of the consequences for selection for parasite resistance, and just generally helping out sheep producers across the country.
I did want to say that you mentioned that you had to shear your sheep this morning when we were talking about scheduling. So you definitely are a farmer or rancher. Which do you prefer?
Well, so I'm on the east side of the country now, so they call them farmers out here. Where I grew up, everybody's called ranchers, so it's just a difference of vocabulary. So a part of my job is also running the sheep unit that we have here, and because running it is so integral into our research program, I get pretty involved in that stuff. And I grew up shearing sheep. My dad taught me how to shear, and then I sheared all the way through college, and that kind of paid for bills as I was going along the way. So it's hard. It's one of those things that it's really easy now. It's easy to do, but it's I still enjoy it at some level, I guess is what I'm saying.
And I believe you said I read you raised Rambouillet, and which is that a wool or a meat sheep or both?
That's a wool breed, classic Western range wool breed.
Yes, and Western range is quite a bit different. We actually just came through the Central Valley from Oregon down to San Diego, back down the 5, and that's quite a different environment than it might be in the East Coast.
So, right, and when we're talking about Western range sheep, most of those sheep were developed in collaboration with the National Sheep Experiment Station in DuBois, Idaho. So, when I talk about Western range sheep, I'm talking about those Rambouillet, Columbia, Corriedale, Polypay, those type of sheep that graze a lot of federal land and BLM land, going down from Idaho all the way down to Southern California. So.
Yeah, people don't really realize how much BLM land is out there and how sheep just kind of can run, and it's not an intense operation necessarily. It's just out in the range, correct?
Right. So we refer to them typically as extensive operations. So they're not running 40, 50 head. They're pushing 20, 30,000 ewes through there. So a little bit different.
So but I'm assuming too that these sheep have to be a little bit more self-sufficient if they're just ranging in a broader area.
Yeah, I mean, you're moving the animals to feed, as opposed to what we're more accustomed to is bringing feed to animals. So these are nomadic flocks. They graze in those different lands, and if they've got to cross some other lands, they've got to get trailered across there. So I grew up in an area in California where we'd constantly have sheep coming through that area. So there's a lot of large bands of sheep that would come through and hit those alfalfa fields in the winter, December, January, those type of, and lamb in that area before they're moving on.
Now we just came from Oregon. I mentioned that we were up there with Martin Daly and Joy, and Martin has quite extensive experience in raising sheep, but he used to raise merinos down in a place called Dixon, which is close to Davis, California. Yep. And again, it's that hot, dry area. He said that he tried to take his merinos up to Oregon and that his line just did not make it up there. It was too wet and dry. So I would assume that if you had sheep experience in California, does that translate to your move back east? And is it the same kind of thing, raising sheep back there?
It's different, right? So you've got a different environment. I grew up raising more show type sheep. They weren't necessarily commercial or range animals. So that's, I mean, we only had about 100 ewes at home, and it was more for the purpose of getting us out of the house and getting us on the road and doing those kind of things. Raising sheep is just kind of raising sheep, and you adapt to your environment, right? So some of the stuff that works in California is not going to work out here, and vice versa. And the type of sheep that are out here versus what's in California, um, it's pretty diverse. I would say, in the 1800s, there was a lot of merinos in this part of the country, in Virginia, West Virginia, Pennsylvania. That shifted over time. It's easier to raise those animals out in Nevada, Arizona, California. Uh, they fit their environment a lot better. So we tend to have a lot of farm flock type sheep out here, Dorset, some polypace, but we see a lot of infusion of Katahdin hair sheep in this part of the country.
Now your study is on, I'm going to let you talk about it in just a minute, it's the Barber Pole worm, but that also is something that responds to wet conditions. So I can see why back east you might have quite a lovely study group instead of out west where it's really dry.
Sure. So it typically hits this part of the country. There's going to be a paper that will come out here shortly that talks about parasite prevalence in the Intermountain West areas, Montana, Utah, Wyoming. I've done studies with friends of mine who are high school ag teachers in Central California, and while we get more rain for free, right? The irrigation that goes on in California can also severely impact. You can have quite a few parasites out there. So in Northern California, they do a lot of flood irrigation. Central California, it's still either center pivot, flood, or kind of row irrigation, but those kind of things are still going to allow that parasite to survive. So while it's hot and dry, which is correct, at the surface level on the ground on irrigated pastures, you're still making a humid environment for that parasite to survive.
Yeah, I had the feeling in California we're going to move a little bit from the flood irrigation to the drier stuff just out of necessity, the way that our climate is going. So very definitely
Sure.
to be changed. Okay, so just a couple terms. I would love it if you would clarify for the layman. One of them is ruminant. Tell us what a ruminant is.
A ruminant is an animal that has a compartmentalized stomach. So their stomach has 4 large compartments in it. So a ruminant has the ability to take forage resources or grass and turn that into and actually utilize that grass as a source of energy and protein. So our stomach's not set up to do that. So there's 4 compartments to the ruminant stomach. There's the rumen, which is kind of where microbial fermentation occurs in that system, the reticulum, the omasum, and the abomasum. So the 4th compartment, or the abomasum, is the site of chemical digestion, and that's where that's where that section of their stomach looks the most like ours. And if you want to think about it, that's the one that's akin to what we have. So it's an acidic environment. That's where you're going to get a lot of breakdown of those different feed components there too. So the bugs and the rumen microbes break down a lot of the starches and cellulose that are in plants, and then that is utilized by those bugs. And some of the bugs actually end up feeding the animals through microbial digestion and breakdown of those microbes as they exit the rumen.
Yeah, so a whole little ecosystem going on in whatever body there happens to be. It's not just one thing.
Correct. Right.
So are sheep and cows are both ruminants? Do they kind of work the same way?
Yeah, there's economies of scale, I guess, is the other part of it. I mean, that's past that. There's not a whole lot of similarity between the species in terms of the way they behave, act, reproductive cycles. Those are all different between those species. So, yeah, they're ruminants. So are deer. So are goats. There's a lot of ruminants in the world that utilize plant resources for food. So,
excellent. Okay, so So with sheep, does their gut work the same way that it might in other animals in terms of the length of the gut and how acid it is and how things flow through it?
Yeah, yep. So like I said, there's economies of scale, right? So there's the rumen is not as big in a sheep as it is in a cow, right? So that makes sense. You look at a cow's got a 40-gallon rumen. A sheep, a mature ewe, is going to have about a 20-gallon rumen, right? So those kind of things, you've got differences. There's going to be differences in terms of length of the small intestine and large intestine, those type of things. But the basic functionality of that stays pretty consistent between the species. So between cattle and sheep, the pH level of the abomasum is pretty constant.
Okay, that's great. Now, all this is going to come back to the worm here in a little bit,
Hmm.
as I'm sure you know.
Yep.
So I'm going to slaughter this in Latin. So I'm going to have you correct me, but I'm going to say Haemonchus contortus, the barber pole worm. So it's pronounce that correctly and tell me what that is, please.
Okay, so Haemonchus contortus, that's just my California pronunciation of it. And you'll hear people call it Haemonchus. It's fine. So we just call it we end up in our lab, we just call it HC more often than not. But Haemonchus is a blood feeding parasite. So this parasite is has different stages of development. So if you think of it, the easiest way to explain this to people is to start coming out of the animal. So if there's eggs that are coming out of the animal in their feces, those eggs will develop through 3 larval stages in the environment. And for that to occur in a fairly steady state, there's some environmental requirements that have to be met. There's got to be humidity and typically heat. So we're talking about, if you want to think about the perfect environment for Haemonchus to live in, think of the Caribbean, right? It's hot, humid, and warm all day long, all year long, same temperature. You don't get big fluctuations and those type of things. So out on the pasture, those eggs are going to develop into larvae, and the larvae will go through 3 stages of larval development on pasture. So from the time the egg is shed to the time that you have what are called 3rd stage larvae, or the infective stage, is anywhere between 7 and 10 days on the pasture. I can recreate this in a laboratory setting and get that done in as short as, typically it's about 7 days. It can happen earlier. It just depends on environmental conditions. So those larvae are out there, and just to give you an idea, they're about 7/10 of a millimeter in length. So they're not very long. I don't want you to think about them moving like an inchworm or moving like a large worm. They don't have enough body or mass to really do that. So they actually are more swimmers than they are crawlers. So they utilize moisture on plants. So they will ride in the dew as that moisture moves up the plant and down during the day. And that kind of, so if you think about when dew is high on plants or on grasses, it's typically the same time when animals are grazing, right? They're grazing in the morning and at night, maybe a little bit during the day, but nothing. It just depends on the ambient temperature outside. So they're going to move up and down. Sheep are going to consume, or any ruminant for that matter, sheep, goat, cattle, they will consume those larvae, and the larvae will go with the digesta through all of the compartments of the ruminant system, and then stop when they get into the abomasum. So once they reach the abomasum, those worms will, and this was best described by a fellow back in the early 1900s about this, the way this works. So he described this as making a small blood droplet on the inside of the abomasum, and then that larvae will wrap around that blood droplet and use it to feed on. So by the time they get into the animal to about 14 days later, they're going to go through 2 more molts and then become adult worms. So they're going to go from being about 7/10 of a millimeter. Adult females can be upwards of 2 centimeters in length, and going to do that in a period of about 14 to 21 days. Once those adults are mature, you're going to have male and female worms. They will mate. The females will produce eggs, and females will produce somewhere around 10,000 eggs per day. that are fertilized eggs that are shed out into the digestive tract and then follow the animal out and get dropped on the ground. While she's doing this, these worms are going to take in about 50 microliters of blood every day, right? So that's kind of daily consumption of blood from those animals. So the Haemonchus at the end of its mouth has what's called a lancet. So unlike other parasites, like if you think about hookworms and dogs, they have no hooks around their head. There's no mechanism for them to hold in place. So Haemonchus will produce a lancet and then pierce the side of the abomasum, and then just kind of open their mouth and let blood flow into their body, right? So they get that barber pole appearance, or candy cane, or anything that's red and white twisted together, because their intestines and ovaries are kind of twisted together. So the ovaries are white, the red part is their digestive tract, and that's why adult females, or that's why that worm gets that barber pole name. So 50 microliters to make this relevant. If you've ever tried to hold a drop of water on your fingernail, that's pretty close to about 50 microliters, right? So it's not very much. Multiply that by 1,000 worms, now you're talking about 50 milliliters of blood. And that's about the size of a Dixie cup, right? So if you've got a small lamb, 50 milliliters is a lot. You got a 250-pound ewe, 50 milliliters isn't anything, but this is cumulative. This is happening on a daily basis. So you're taking away nutrients that could be used for maintenance or growth or lactation or a lot of other things when that animal constantly has to regenerate red blood cells due to infection.
Yeah, I know that they have that same issue. We have it with our chickens. We had an outbreak of mites that was brought in by birds, and you can barely see the mite, and yet the chickens are getting anemic because there are so many mites, and they multiply at such a fast rate that the chicken can't produce blood fast enough to keep up with the mite, and it can actually kill them, exsanguinates them.
That's effectively what happens in acute haemonchosis. So when you have enough larvae or enough worms in there that are feeding on blood, they will die from exsanguination.
So is that how you find, do you find the issues with like for instance, with a sheep, you know it has anemia, and so would barber pole worm be the first thing you would test for?
Well, so the first thing that we figure is that, particularly on the eastern part of the country, we use a method of selective deworming. So what we do is we check the eyelids and the color of the mucous membranes underneath their eye. So this is a system called FAMACHA, or FAMACHA. It was developed by Fafa Mullen, and it's Fafa Mullen's chart, or FAMACHA. So this scores the anemia of the animals from 1 to 5, 1 being very red, 5 being white. So this gives a producer an idea of how anemic an animal is, particularly a grazing animal. And then during the summertime, we assume that most anemia is probably parasite derived and not nutritionally derived. So that can kind of give us an idea of how to treat animals, when to treat them, those type of things. In terms of distinguishing whether it's Haemonchus or some other parasite, there's no real way to do that other than looking at the pathological effects of what you've got. So if you've got an animal with anemia and they have a high fecal A count, there's a pretty good chance that that's Haemonchus. But that doesn't mean that just because an animal has a high fecal A count that it is only Haemonchus. There are other, what we call trichostrongylid parasites. So that would include Telodrossagia, which is also an abomasal nematode parasite or worm parasite, Trichostrongylus, which is found in the small intestine, Cupuria, which is found in the small intestine. And they all have the same egg shape. So when you look at them on a fecal egg counting slide, or you look at them under a microscope, you can't really tell the difference between them. So the only way to do that is to culture the eggs, get them to become larvae, and then measure the length of their tail.
Wow. Oh, now I could see how that might be a real problem, especially if you have, if you're trying to do parasite control by medication. I know with the chickens, it's one of those things that you have to be specific in terms of what you're trying to fight with the parasites, and they become resistant over time. So I can see with sheep, if you're aiming for one thing and you have to constantly bombard them with whatever medication you think might work, that you need to go outside the box and find something that more naturally let them compete over time with parasites, correct?
Correct, but I still think that treatment plays a role in a strategy of parasite management. And I don't think that our aim is to make sheep sterile of parasites. Sheep are going to be exposed to parasites. What we don't want to have happen is that the parasite becomes so overwhelming to their system that they don't perform for us.
Excellent. Okay, so this actually leads into why you're studying the St. Croix sheep. Tell us about that.
So the St. Croix sheep originated on the island of St. Croix. They were brought to the U.S. by some researchers out at Utah State in the early 70s, and they started doing work with thinking about incorporating St. Croix. So if you guys are out in California, you'll see a lot of Dorper St. Croix crosses or Dorp Croix out there. Southern Oregon will have those, and they'll have some in Texas. The St. Croix is also used as one of those progenitor breeds to develop the Katahdin breed, which is currently the most popular breed in the U.S. They've got more annual registrations than any other breed, far surpassing Suffolk or any other breed in the U.S. So they're kind of a trendy breed right now, but they have parasite resistance, and they also have the other part of not having to shear them. So the problem with Katahdins and kind of understanding immune mechanisms is that there's so much variation in how one Katahdin is going to perform versus another. So we went right to the source. And this started with my PhD work down at Virginia Tech, because they had a group of Barbados blackbelly ewes and that had St. Croix and just a little bit of crossbred of the Caribbean breeding on those animals. So we started doing work there, and once I came to West Virginia University, we've expanded that and kind of dove deeper into immune mechanisms and how these sheep respond and what makes them different than other breeds.
So what does make them different than other breeds in terms of how they process the Barber Pole worm?
What we have at WVU is we have a system where we can create sheep that are naive to parasite infection. So they're born in an environment where they're never going to be exposed to a worm. So experimentally, what we can do is look at what happens at the first time when those animals are exposed to Haemonchus. So when they're exposed to Haemonchus the first time, the St. Croix sheep will clear the infection by themselves somewhere between 5 and 6 weeks after infection. So we typically, as a part of our experiments, will give lambs about 10,000 Haemonchus contortus larvae, and their fecal egg count will go up to about 3,000 or 4,000 eggs per gram, and then a couple weeks later it'll bottom out.
So it's like cycling a fish tank. They get certain levels come up and then other ones go down again, or is it they're just done?
Yeah, they don't go up and down because these animals are in an environment where they can't be reinfected by anything picking up off the ground. This is all in controlled housing and barns where we do that. So they're not out on pasture doing this. Now, if we compare those to Suffolk sheep that are raised in the same type of environment, the Suffolk sheep will actually get a higher fecal egg count than the Croix, even though that they're given the same number of larvae, and they will have a static infection. They will maintain a high level of infection and won't clear it the first time. So we usually will drug treat those animals, knock out the infection, wait a couple weeks for the immune system to kind of recover from that, and then go back and reinfect them. When we challenge, it's called challenge infection, when we challenge the St. Croix, their fecal egg count may go up to 100 eggs per gram, and then it's gone, right? When we challenge infect the Suffolk, they go right back up to 3,000 or 4,000 eggs per gram. So the first thing that we're seeing is failure to develop protective immunity. So for whatever reason, the Suffolk sheep they require a lot more exposure to Haemonchus before they will actually develop an immune response that is effective to clear the parasite. And we know that mature Suffolk ewes have have a fair amount of resistance to this, right? But it's not a function of genetics, it's more of a function of exposure. So those animals were exposed to it many times, have ultimately developed immunity based on multiple exposures. But if you've been around sheep for any length of time, you'll know that these sheep will find a way to kill themselves. And we don't want to let a worm infection be one of those sources or one of those options for them.
Now, this is fascinating because I would think that this says to the breeder, oh, if one of two things. One, if I've got a problem with barber pole worm, which it's my understanding from all the sheep people that we talk to, this is a major issue for them economically and in terms of management. Is that correct?
Correct.
Yeah. So I mean, this can have a huge impact on how profitable a farm is or not in getting rid of this type of thing. And again, medications are not necessarily bad, but you don't want to have to keep bombarding your sheep with this if there's a natural way to take care of it. Would
Correct.
you say that you might find, as a result of the things that you found in your study, do you maybe run St. Croix sheep through your pastures first before you run any other animals? Or do you mix a little of that in with your other animals? And again, they're a hair sheep and not a wool sheep, so I can see how that might be bad in the long run for your fleece production. But how might you use this practically?
So how does it use practically? That's a great question. So the information that we're getting from the St. Croix can kind of sometimes be egg-headed academic information. So it's not going to give you a whole lot of practical knowledge right out of the gate, right? You're not going to get Western range producers to start crossbreeding St. Croix sheep. Not going to happen. And alternatively, you're not going to have the people buy flocks of Croix to graze before the flock that they really care about, because the availability of St. Croix sheep across the country is fairly low, right? The other part of it that comes into play with this is that St. Croix sheep just by themselves, while they survive a parasitic infection, they're great moms, they're great sheep in general. When you go to market these animals on a commercial setting, they're not going to be as profitable. You're not going to get as much for them sold at a sale barn as you would Suffolk crossbred lamb, right? So there's some inherent issues with that. I think that what we're looking at with this is kind of some of the basic biology of why this happens, right? And this can be implemented as a part of a genetic selection strategy. And that's what the Katahdin breeders have really kind of grabbed a hold of, is how do we use genetics to control parasitism within our flocks? And one way to do that is really kind of focusing on selection for animals with low breeding values for fecal egg count. So a breeding value is simply an animal's genetic merit for some trait, right? So it kind of excludes environment. It's what is their genetic merit for that trait. And an animal has a low or a negative breeding value for fecal egg count, that means that they're going to be more resistant and that their progeny are going to pick up half of that breeding value, right? Makes sense?
Yes.
So they're going to pass on half of their breeding value onto their progeny. And in fact, when you use rams with low fecal egg count breeding values, you get lambs that have low fecal egg count breeding values. So you can select for this immune response. And that's effectively what you're doing when you select for fecal egg count reduction, like they do through the National Sheep Improvement Program, is to utilize those values to identify animals that are going to be a better, it's kind of an insurance policy against parasitism. It's not the full answer, right? So this, in combination with some rotational grazing, with selective deworming, are all a part of a systematic approach to managing parasites within your flock.
Now, I mean, that's an excellent point, that this is just one of the tools, but I can see when you select for trait over time, you select for one thing and you potentially lose another. So I could see that maybe you would have to use this in an extremely scientific way if you truly wanted to select for this trait, because you have to do genetic testing, do you not, on any animals that you want to see that they have this, or no? Is there a simpler way?
Yeah, no, this is pretty simple. So West Virginia University, Texas A&M, and Louisiana State University are all a part of what's called the American Consortium for Small Ruminant Parasite Control, with a number of other universities. But these three institutions will actually do fecal egg counting for you at a fee for $5 a sample. So you submit a sample to one of these institutions that's closest to your location, and you will get data back within a week that you can use to submit with the rest of your data on your animals so this isn't like genetic testing in terms of submitting a blood sample. It's genetic testing by submitting numbers, by submitting information. So the things that we're submitting along with fecal egg count are weaning weights, birth weight, birth type, ribeye area, depth, fat thickness, scrotal circumference. So this incorporated into a selection strategy can have an overall benefit for your flock. And we've demonstrated that through our collaboration with the folks at Virginia Tech and using their Katahdin flock and actually divergently selecting rams with either high or low fecal egg counts and randomly mating them to Katahdin ewes. The lambs that come from low fecal egg count sires have low fecal egg count breeding values themselves. So it's a moderately heritable trait, so meaning that you can make improvement on it in a relatively short period of time.
So if we do have farmers that we meet that say, oh, that's a great thing, does that apply to wool and meat producing sheep? And how do I find out more about that? What would you recommend?
Sure. First thing that you want to do is go to nsip.org. So Rusty Burgett is the coordinator for the National Sheep Improvement Program. There are information about wool breeds. The National Sheep Improvement Program works with a group out of Australia called Lamb Plan, and they will calculate these values for you, right? So you submit all the information, and Rusty can help any producer do this and then get those data back and then start thinking about how do you incorporate these data into your flock management scheme.
It seems like an excellent resource to me. Now, one thing I did want to ask, because I understand that sometimes if pasture management is a part of what might help sheep to be healthier in attempting to naturally combat some of these things and increase immune response, what kinds of things in terms of protein or nutrition do you find that would naturally help sheep to combat this barber pole worm?
So the first thing that we can talk about with grazing is that on pasture or where these animals are going to pick up these worms, right? So that's where that's going to occur. Goats tend to be more browsers than grazers, but if you change their environment and take them out of a browsing environment and put them on irrigated pasture land or fenced in pasture that doesn't have any plant resources to browse, you're now changing them into grazers. So how long you're on a pasture, how long you're on a paddock within that pasture, those kind of things are going to affect the exposure of those animals to parasites. So some people will call it avoidance grazing. So that doesn't mean that you need to move the animals every day, right, and not utilize the pasture resources that you have. You have to think about how do you best manage the animals and also best manage the land. So I think that sometimes when we start raising livestock, we forget about we're also grassland managers as well. And that has to be an important component. The second part of all of this stuff is understanding what happens nutritionally to an animal when they become parasitized. You've now changed that animal's basic or basal requirements, nutrient requirements. So if you've got a nutrient requirement for an animal that is not infected with parasites, that's going to be different than an animal that is infected. So understanding right off the bat that anytime I've got a sheep grazing on pasture, they're exposed to a parasite infection. And that whatever it says in a sheep book about how much feed they should be getting on a daily basis has to be altered to compensate for the parasitism those animals are experiencing. So my professors at Chico would call it the difference between the science of feeding and the art of feeding. And there's an art to this, and this is where the management side comes into this. Now, what we've done over the past few years in West Virginia is to look at the importance of both protein and energy nutrition in helping an animal experience the largest amount of immune response to a parasitic infection. So one of those ways to do that is simply through energy. So energy is a rate limiting nutrient in terms of immune response. If you don't have any glucose and you don't have any of that, any energy in the diet, then you're not going to get immune response, period. But then right after that comes everything else. So everything your immune system is doing and generating is all protein derived. So if those animals are also protein deficient in pastures, then that will also affect those kind of things. And then we also know that protein comes in a variety of different forms, and how that is degraded in the rumen, or if it bypasses the rumen, those kind of things will affect the availability of proteins and which specific amino acids are available for digestion. So you'll hear ruminant nutritionists talk about the rumen as kind of being a black box. What goes into it and what comes out of it are two different things, right? It's not like a pig where you can put feed into one end of the pig and know exactly how everything is broken down, because there's no microbial fermentation that's occurring in that digestive system, or not nearly as much as what's going on in a sheep. So one things that we've been doing is utilizing rumen protected protein. So things like adding fish meal to a supplementation of lambs that are being grazed. I think that sometimes when we test our pastures, the values that we get back, we get really excited about. And you have to remember that that value that you get back on that test is everything that you sampled. But then you also have to look at what you sampled versus what the sheep is consuming. And those two things are often at odds with each other, not very equivalent. So by supplementing lambs that are grazing, and this is contrary to everything that I was taught as an undergrad or as a young man raising sheep, is that sheep run on grass. You shouldn't have to supplement them. You shouldn't have to do anything like that. Yeah, well, then you get into the art of raising animals. And you know that those animals that are parasitized, they're already protein deficient. They are going to require more sources of energy and more sources of protein that are concentrated. So we use a strategy of supplementing lambs at 2% of their body weight, and then we add fish meal to that. And once we add fish meal to that, not only do we get an increase in gain on those lambs that are grazing pastures, we also get a reduction in fecal egg count, which is contrary to what happens when we supplement with a mixture of corn and soybean meal. And that's kind of more of a common mix. That particular mix, when soybean meal is the only protein component there, then what you get is you get a resolution of the pathological effects of Haemonchus infection. So those animals won't get as anemic, but they're still going to have high fecal egg count. So basically, by giving them that supplement, all that supplement is doing is going to mask the effect of the worms on the animal's anemia. But you're not going to get those. Ultimately, we still have to get a product out of these animals, and we still have to make sure that they're as healthy as possible and that we're generating a healthy product. So by adding some rumen protected protein in there, particularly through byproduct feeds. So if you think about corn gluten feed, which is a byproduct of making high fructose corn syrup, or fish meal, which is readily available everywhere across the country. I mean, it's around with a lot of the aquaculture enterprises that are going on. Those are all sources of rumen protected protein that we have published data on that indicate a reduction in fecal egg count and an increase in growth.
Now, I would think also that with all that you just said about nutrition, especially with a wool product, we learned recently that when we spoke to the wool producers, that they had a bad rain year and their grass was completely nutritionally different than they otherwise might have been. It got too soggy, and so it really impacted something that's dependent upon a consistent growth and a consistent nutritional profile, and their entire fleece could have been completely ruined because they had 1 month of bad nutrition. So I can see how when you have a product like wool constant nutrition, especially with something like a worm that can impact the amount of uptake of nutrition that the animal has. Whether the animal is eating it and whether the animal is getting the benefit of that are two different things often.
Right. So trying to avoid peaks and valleys in nutritional levels is important for growing animals, for lambs that are growing, or if you're trying to grow fleece, constant fiber diameter, kind of consistent strength in that fiber going from the skin side all the way out to the end of the fleece are critical things. And I can see this when I'm shearing animals. I know that if there's, I mean, if you've sheared sheep after a used lamb or has weaned a lamb, you know you're going to have breakpoints in that fleece right at the skin level when you shear those animals after weaning. So those things we know will pick all those kind of, you can pick that up in the fiber, and the fiber is going to be a telltale sign of what's happening in those animals. But sometimes with growing lambs, you don't have that opportunity to see the effect of that. So if you've got real heavy rainstorms that come through for a month or so, you get grass that's washed out. Remember that if an animal's eating a pound of grass when it's really wet outside, it's going to reduce the amount of total grass that they're getting, right? In terms of a dry matter basis, they're not getting as much from that. So those are all things to think about. So that's why I think that supplementation, even when animals are out on pasture, is still a critical component of maintaining productive states in livestock, whether your focus is on wool or lamb.
This is absolutely fascinating, because I would have assumed that the resistance to the barber pole worm was very much a physiological thing. Maybe it's the acid in the sheep's stomach and the St. Croix sheep's stomach that causes that. But it's immune response, and a healthy immune response, it seems to be what you're saying.
Correct. So the acid, so the worm, when it gets into the stomach, regardless if it's in a Croix or the Suffolk sheep, or what we use as our susceptible model breed, what happens in there is that once the worms get to a certain size, they produce an excretory secretory protein, and that will actually buffer the pH in the abomasum. So that's one of the things why you don't get as good a nutrient digestion in Haemonchus infection, is because those adult worms start changing the pH in the stomach, right? So now, that's why the other reason is that you don't see diarrhea with Haemonchus. So Haemonchus doesn't generate diarrhea, but will change the color of the stool because some of the blood that's leaking out of the abomasum.
This is fascinating. So are you going to, now, is this an ongoing study, or is this a one-time study that you've done?
No, this is a 30-year, this is what you make your bones with in academia. So it's the way I was mentored was that you find your one thing that you're going to do for 30 years and you spend your time doing that. And then when you're done, you hopefully have made some contribution to science and agriculture, and that's what you get to retire with, right? So we were looking at a lot of different things with this. Immune response, the way that cells behave differently in one breed versus another, some of the genetic mechanisms that are underpinning this difference in immune response. Really, the St. Croix sheep the best way to put it is they kind of have a kitchen sink response to larvae. They unload everything: antibody cells, cytokines, inflammatory mediators, it's everything, right? And when the Australians really put a lot of selection emphasis on fecal egg count reduction, what happened was they started building animals that developed such a polarizing immune response that it actually injured the animal. So it kind of went towards a autoimmune disease state as opposed to a protective state. What's interesting about the Croix is they were never selected directly. They were never artificially selected for fecal egg count. These animals were just dropped off by merchant ships that have been passing through the Caribbean since man started moving over from Europe and Africa over the continental US or North America. So these animals had to survive in this environment and had to develop a mechanism to survive. And that's what they do. I put them out in an infected area, they're going to be just fine. You're never going to have a worry about that.
Well, I have to say that when I started this conversation, I thought, okay, this is broader applications because other species get this. Cattle get Barber pole worm infections and things like that. It's my understanding, not as much as sheep, but they can cross to things like cattle, correct?
Yeah, so cattle don't really get Haemonchus contortus that much. They get another version of Haemonchus called Haemonchus placii. So, and Haemonchus placii you just don't see in sheep very much. So Haemonchus in a beef cow, she's going to chew that thing up and spit it out. So usually in this part of the country, we really recommend to a lot of small ruminant producers, you want to get Haemonchus out of your pastures, graze cattle there, or take a hay cutting, right? So any way to dry or desiccate. So if I leave larvae out on a glass slide, and let's say that one of the students were doing some microscopy and they forgot the worms were on there, 30 seconds, that little pool of water is going to dry up, that worm's dead, right? So if you can find any way to desiccate that pasture without making it completely dry and not have any moisture in it, it's a great way to do that. So we can clear out a pasture by taking hay cuttings. A mixed species grazing is another great approach to managing this. Cattle graze in different areas than sheep, but cattle can also handle that infection, and actually it doesn't really establish that well in beef cattle. So beef cattle are more susceptible to parasites like Ostertagia, which is an abomasal dwelling parasite, and Cupuria, which tend to affect them a little bit more than Haemonchus does. At least in North America. In South America, Haemonchus placii will tend to affect beef cattle a little bit more.
But as you're describing this kitchen sink response that the St. Croix have, I
Mm-hmm.
went from, okay, sheep and maybe small ruminants and cattle to all of a sudden, holy cow, if you could figure out how you get the kitchen sink immune response, think about what that might do potentially in the long run for people, for any animal. If you can figure out how to stimulate that immune response, you can retire in St. Croix.
Yeah, so that's the trick with all of that. One of the things that we've done is done some different additive feedings that are immunostimulants. Um, it's just not a clear-cut way of how you get that done. What we are working on now is one of the immune products that's generated by sheep in general to Haemonchus, but just more so in the Croix is a component called interleukin 13. And that interleukin 13 does is it has two effects. It paralyzes larvae. So when I take interleukin 13 and I put it in with larvae in the laboratory, the larvae become paralyzed. And all of our drugs used to treat parasitic infections have the same, they're just paralytics. And that's what they do. So if something's paralyzed and it can't move and it's in the digestive tract, it's going to go with the flow, right? So it's going wherever the rest of the food's going. And that's the way dewormers work, is they paralyze larvae through different mechanisms, right? So they don't actually go in and kill the larvae, they paralyze them. So we can tell that they're paralyzed, but they're not dead by looking at their ability to generate energy in the form of ATP. So we measure ATP in those larvae after they've been exposed to IL-13, and they're alive, right? They're not dead. So that's one thing. The other thing that IL-13 does is it causes, at least in mice, it causes a weep and sweep response. So it loosens up the tight junctions in the gut and allows fluid to cross that barrier. So it kind of induces diarrhea, right? So if you're not going to paralyze something, the other way to get it out of your system is to increase the flow rate, right? So if
Flush,
you
yeah.
got transient, right? You got transient diarrhea that happens with this. That's what IL-13 is going to do to the digestive tract. So the great thing about, and what we're kind of trying to figure out, is how can we administer IL-13 in the digestive tract to get both of these responses. So it's both a larval paralytic and it's going to induce luminal flow and kind of gut contractility. So that's the weep and sweep response. So it's stuff coming across the lumen, and then it's going to increase the muscle contractions. You know, when you're sick and you're not feeling well, your stomach hurts, right? That's the contraction. And then you have luminal flow, so all that water's leaving you. They tell you to drink a lot of water when if you have diarrhea. Same kind of thing here. So if that's doing that a little bit in those animals, that might be another way to address treatment. Because you're right, a lot of our drugs aren't working anymore for managing parasitic infections. Even from a single dose standpoint, ivermectin doesn't work very well in some cases. Any of the benzimidazoles like Safeguard or Valbazin or Panacure, those aren't working very well anymore.
And the externals too, correct?
What's that?
The external applications are, you know, the oh gosh, I'm completely blanking here. Not promethazine.
Permethrins.
Permethrins,
Yeah.
yes, thank you.
Mm-hmm.
Those are becoming less effective sometimes as
Right.
well too, correct?
But these ones, I mean, this is documented lack of efficacy. So when you've got single, double, and triple dewormer type that is not effective, period. We got producers that are dealing with that in Australia, New Zealand. We're starting to see this. There's some folks that are heavily involved in the American sheep industry that are in South Dakota. Their family's been raising Rambouillet sheep there for generations, that are losing sheep to parasites, and they've never lost them like that before. So a little bit of climate change and the northward prevalence of Haemonchus. Haemonchus doesn't really like cold weather very much, but if it's a little bit warmer up there now, you're starting to see Haemonchus more in England and Northern Europe where it just wasn't there before. So either the parasite's adapting or the climate shifting a little bit, that's going to allow those worms to survive in that environment. But I mean, we're talking about massive losses, 200 lambs dead from parasites, and they dewormed them, right? So this kind of gives producers, it's been shaking producers up over the last probably 5 to 10 years, made massively in the Western US. So I think that folks out West are starting to realize that this is a larger problem and it's not just isolated to Eastern sheep production.
Yeah, major economic impact. And Western Australia comes to mind with all those the wetness
Yep.
there. They have a big problem with these types of things in the Barber Pole worm. This is fascinating. I don't want to take much of your time because we're bumping up on the time that you promised us. But I have to tell you, this is just, I can see not only agricultural applications but human applications for your 30-year study. If people wanted to follow you and find out more about this, where do they go?
My website is bowdridge.davis.wvu.edu, or you can just type my name into a search bar and that page will come up. There's information about what we do in the lab. We work with St Croix sheep, Texel, and do a lot of genetic work with some Katahdins and our collaborators down at Virginia Tech.
Wonderful.
There's another website that has a lot of information for producers and managing worms. It's wormx.info, and it is the website for the American Consortium for Small Ruminant Parasite Control. And there is fact sheets, places to get FAMACHA cards, how to get FAMACHA training. All of those things can be found on that site. How to manage this with genetics, nutrition, dewormers, advanced parasite control strategies can be found there.
I'd actually been to that one before I spoke with you. I now feel like a sheep person.
Good. There you go.
Well, thank you so much. This has been absolutely wonderful.
You're welcome. Anytime.